Good evening, students. Today's topic for this video is pollination. This is for class 12, chapter 2, flowering, reproduction in flowering plants. So pollination is not a topic that we don't know about. It is a topic that we start studying from class 8 onwards, I guess. So pollination in a very simple manner is the transfer of pollen grains from the anther to the stigma of a flower in very simple manner. But being this topic included in class 12, so we have to go through detailed, we are going to study this topic in a very detailed manner. So pollination, yeah, definitely it is the transfer of pollen grains from the anther of a plant to the stigma of the flower. So this transfer of pollen grain is the basic principle or the basic procedure of pollination. So now pollination can basically be of two types. If I say about the transfer of pollen grain, it can be indirect or direct. Indirect pollination is what I have just spoken about. It's the transfer of pollen grain from stigma, from anther to stigma. That is actually indirect pollination. And direct pollination occurs in only gymnosperms where naked seeds are there, and so they do not have such stigmatic arrangement actually. So what they have their micropylar opening, and from that micropylar opening of the ovule, this pollen grains get entered inside this ovule and get fertilized. So it can be indirect where major angiospermic flower takes place, and in direct in gymnosperms. So now as we know that pollination can be categorized mainly as self-pollination and cross-pollination. So we are going to be studying today in detail about self-pollination. So what is self-pollination? First of all, self-pollination is the transfer of pollen grains from the anther of one flower to the stigma of that same flower or to the flower of that same plant. That is called as self-pollination. That means एक ही plant में एक सेम फ्लावर के अंदर पॉलिनेशन हो रहा है, या फिर उसी प्लांट के दूसरे फ्लावर में अगर ये ट्रांसफरेंस हो रहा है, then this is called to be self pollination. Now there are various types of self pollination. Again, self pollination can be broadly now categorized as autogamy or geitonogamy. Autogamy, the word itself carries two meanings. Self marriage, that means in a single flower, in one flower only. So autogamy में क्या होता है कि एक ही फ्लावर के अंदर this pollen grain get transferred from the anther to the stigma, whereas in geitonogamy transfer of pollen takes place from one flower to another flower of the same plant or genetically similar species. Now these are the two variants of self-pollination. Now we are going to see in detail about autogamy and geitonogamy. Now as you can see, I have made a flow chart to understand pollination. It can be of two types: self-pollination, cross-pollination. Pollination meaning is stigma, transfer of pollen grain from anther to stigma. In self-pollination, it is definitely it is in the same flower or genetically similar flower of the same species. Now it can be differentiated into autogamy and geitonogamy. The first thing, that is the autogamy. Autogamy contains two words like Greek words autos and gamos, that is self-marriage. It is self-pollination which occurs between anther and stigma of the same flower. It occurs in all those intersexual or perfect flowers which either remain closed or where anther's pollen came in contact with stigma by gravity and mechanical means. Autogamy is of the following types. It can be homogamy first. Now what is homogamy? The flowers are open. The flowers are open, or in other words we can also call them as chasmogamous. They are also called chasmogamous. Both anthers and stigmas are closed together. They mature simultaneously with synchrony in pollen release and stigma receptivity. Now, in open flowers, or when we are saying this homogamy, how we are going to understand it? We are going to understand it by taking two examples of two different species of flower. First is convolvulus and second one is lilac, as you can see. Now, how we are going to understand this? In the first flower, as you can see here, in convolvulus, stigma comes in contact with ripe anthers and receives the pollen grains. That means when the anther occurs, when this style grows, anthers are at the position, anthers are already grown. Now when this stigma grows, they grow to such a length that they reach at least to the strands where these anthers are, and now the dehiscence of the pollen grain takes place. That means in the first case of autogamy, like yes, we are studying about autogamy of self-pollination. We were here as convolvulus, the species where we are going to study about autogamy. When the style, this grows up to the length of the anther, the pollen grain dehisces and the pollination takes place. First thing. And in the second case, as in lilac, the style does not grow, and the stigma remains below the level of the anthers. As you can see, I have tried to draw it. The style does not grow. They do not grow up and upreach to the length of the anther. They do not grow. As the anther mature and dehisce, the pollen falls down due to gravity over the surface of stigma. So that is how homogamy or the pollination takes place in these two species, that is convolvulus and in lilac. And so this was about homogamy. Now the second one that we are going to study about under self-pollination after autogamy is cleistogamy. What is cleistogamy? The word contains two Greek words. Kleisto means closed and gamis means marriage. Closed marriage. What does that mean? The flowers remain closed with anthers and stigma lying close to each other. When the anthers dehisce, the pollen grains come in contact with stigma and pollination takes place. That means in this flower, the flower that does not open at all. They remain in the bud condition. We can say very simply, they remain in the bud condition. They do not open, and the dehiscence of this pollen grain to the stigma takes place inside this closed flower. Cleistogamy ensures self-pollination as there is no chances of cross-pollination. Obviously, we know that. It also ensures seed set even in the absence of pollination. What are the examples we are having? We are having balsam, oxalis, viola. Third type of self-pollination is the bud pollination. Anthers and stigmas of an intersexual or perfect flower mature before the opening of a flower. It causes self-pollination in the bud condition, like pea, jute, rice, linseed, etc. So these are the three different types of self-pollination. Now under autogamy, self-pollination, it contains two types: autogamy, geitonogamy. And autogamy, we have studied about homogamy, cleistogamy, and the third one is the bud pollination. Now, what is geitonogamy? Yes. Geitonogamy is a transfer of pollen grains from anther of one flower to stigma of another flower of either the same or genetically similar plant. That means the pollen does not leave the plant. They move from one flower to another flower of the same plant. That is geitonogamy. Example, cucurbita. Cucurbita family exhibit this kind of pollination, which we call as geitonogamy. Now, importance of self-pollination, we can see here. There are few importance of self-pollination. The first one is, it is a sure and economical method of pollination. Of course, we know that it is sure and economical. That means we do not need any kind of device. We do not need manpower. We do not need any expertise to carry out this kind of pollination. Second, self-pollination is employed as fail-safe device. There is lesser chances of failure. It produces homozygous progeny. Self-pollination is used to maintain pure lines. Pure lines, of course, no hybridization, pure lines are going to be maintained. It maintains superiority of the race indefinitely. Superiority of the race, that means the variety that we are wanting, whether it is a grain or any kind of plant species, the superiority is not going to be mixing because of any kind of cross pollination. It is going to be maintained for a very longer time. The plants need not produce large number of pollen grains. Flowers do not develop attractant for flowering external agencies. They are simple flowers. They do not fragrance. They do not exhibit such characters which attract external agencies. Harmful and useful characters are eliminated naturally. What are the disadvantages now? No new traits are introduced. Obviously, self-pollination, since it does not include any kind of crossing between species, no new traits are going to be produced. It decreases variability and adaptability to chances in environment. What does that mean? Of course, it is not going to be adapted well. If it is a pure line, if there is a chance of infection, the pure line will grow indefinitely as it is. There will be no adaptability because there is no genetic manipulation there. It is going to be, it's a harm for that species. Disease resistance decrease with time, obviously. Recessive traits appear regularly as self-pollination introduces homozygosity. Self-pollination introduces homozygosity. What is homozygosity? You remember that Mendel's laws of inheritance when we write capital TT? That capital TT shows us homozygosity. That means pure lines. There is no way to eliminate defective traits. We cannot cross it. We cannot make a capital T with a small t. Yield decreases with time due to reduction in vigor and vitality. So these are the importance of self-pollination. We have studied self-pollination in detail. In the next class, we'll be studying cross-pollination and other topics of Chapter 2. Thank you.